transcriptionally activated state. And as I have said, activation concerns not the removal or the retention of histones, but rather a transformation from a static to a dynamic condition. The DNA of the gene is then transiently available in a naked form, free of histones, for interaction with the machinery for expression, the so-called transcription machinery. And in our work, for the last 30 years, we have focused especially on the machinery for transcription by the enzyme called RNA polymerase II, Roman numeral II. This is the enzyme which, as many of you know, is responsible for the synthesis of all messenger RNA, leading ultimately to the synthesis of proteins. It is distinguished from the other RNA polymerases 1 and 3, whose products are the stable RNA molecules of the cell responsible for protein synthesis. Now, as the first step in the pathway of gene expression, transcription by RNA polymerase II is a focal point of cellular regulation. It is an endpoint of most signal transduction pathways, by means of which communication with the environment takes place. Indeed, it is, as many of you will know, the intricate regulation of transcription by RNA polymerase II that underlies cell differentiation and development. It was with this in mind that we and others sought to identify during the course of the 1970s and 1980s the components of the RNA polymerase II transcription machinery. In our work and that of others, we discovered that there are 5 proteins required for the process of DNA transcription. These are, of course, the RNA polymerase itself and the 5 additional proteins that go by the letter names, the alphabetic letter names B, D, E, F, and H. The polymerase is alone capable of unwinding the DNA double helix and of the synthesis of an RNA molecule, as I will show you shortly in atomic detail. But the polymerase alone is incapable of finding the start site, the so-called promoter of the gene, incapable of initiating the process of RNA synthesis, the process of transcription. It is for these essential functions of promoter recognition and the initiation of RNA synthesis that the so-called general transcription factors B, D, E, F, and H are required. At first, we believed in the mid-1980s, this set of 6 proteins, the polymerase and the 5 general factors, are not only necessary for transcription, but entirely sufficient. At that time, it was believed that this set of protein molecules is capable not only of promoter recognition and transcription, but also of a response to regulatory influences from within the cell and from the environment. Indeed, there were several experimental proofs of that fact published in the 1980s. Proofs that gene activator proteins that turn on transcription interact directly with RNA polymerase and the general transcription factors at a promoter, at the start site for transcription. Nevertheless, in the early 1990s, my colleagues at Stanford discovered a requirement for an additional component. They discovered a requirement for an activity they referred to as mediator that goes between a gene activator protein and the transcription machinery. In 1994, 2 of my postdoctoral colleagues succeeded in isolating mediator in pure form as a 21 protein, 1 million molecular weight complex. Even then, it was thought by many mediator which we first isolated from baker's yeast was unique to that organism, and that the direct action of regulatory proteins upon the transcription machinery was the predominant mechanism in higher cells, including human cells. It was only at about the year 2000 that we and others could demonstrate the existence also of human mediator. By now we know that mediator is conserved across all eukaryote organisms. The next slide serves merely to emphasize that point. All 21 proteins of the first isolated yeast mediator have direct counterparts in metazoan organisms, such as Drosophila, the fruit fly, and in human cells, and also in plant cells. We have only just begun to understand the mechanism by which mediator performs its essential role. The points listed on the next slide have so far been established. It has emerged that mediator is not only required for regulation, it is required for all detectable transcription of all RNA polymerase II promoters in all eukaryote organisms. Especially in human cells, it could be shown mediator interacts directly with every transcriptional activator protein. As I will show you later, we could see directly mediator interacts with the enzyme RNA polymerase II. It is in this way that mediator communicates information from DNA in the genome, regulatory elements such as so-called enhancers, to the start sites of genes, the promoters. Mediator is commonly referred to in the literature for the reason I have just explained as a co-activator. It assists the role of the gene regulatory proteins that turn on transcription. The studies in yeast, especially by genetic analysis, demonstrated equally a requirement for mediator for the negative regulation of transcription, for turning off genes. Mediator then is a co-activator, also a co-repressor, and then, as I have indicated, a general transcription factor, all in one. So by way of summary, mediator receives and processes multiple inputs of regulatory information, and then transmits an appropriate regulatory signal to RNA polymerase II at a promoter in the entire range of eukaryotes, from unicellular creatures like yeast to humans. Just to add some color to this picture, I'll give you some examples of the diverse roles and emphasize thereby the importance of mediator. So, for example, mutations in mediator subunits prevent flowering in plants. Another example, in more than half of all colon cancers, a particular component of the mediator, one of the 21 subunits, is present in a superabundance. And by diminishing its level, one can restore cells to the normal state. And yet another interesting example. A particular component of the mediator, different from the one that I have mentioned in the case of colon cancer, occurs in 14 different types in the organism Candida albicans, which is a pathogenic fungus. And it is because of that diversity of types of the one of 21 subunits of mediator that Candida acquires its virulence. So then the machinery for transcription consists of nearly 60 proteins that assemble in a giant complex of more than 3 million molecular weight, larger than a ribosome, at every start site of a gene prior to every round of its transcription. Francis Crick, whose name will be known to many of you, is supposed to have said, if you wish to understand function, study structure. The challenge in this case is the size and the great complexity of the structure. It was for this reason that we began in the 1980s, so about 30 years ago, our pursuit of the structure of this machinery by focusing first upon the enzyme RNA polymerase II. Seems to have lost the connection. Is there by any chance an extra pointer? Does anyone carry one when you've got it? As I said, we began with RNA polymerase II. Itself an assembly of 12 proteins with a mass in excess of 500,000 Daltons. It was a challenging, a formidable object of investigation at the time. The largest molecule that had ever been solved by structural methods was about 1/10 the size. There were no methods available in the 1980s for investigating a structure of this magnitude. It was nevertheless, in retrospect, a most fortunate choice. We could not have known at the time, had we begun investigating the structure of one of the smaller, simpler general transcription factors, we would have been unsuccessful. Because, as I will show you in one example later, these molecules adopt their defined structures only in the complexes that they form with RNA polymerase II. This enzyme, polymerase II, this assembly of a dozen proteins, is the platform upon which all the other components are assembled. And as I believe you will appreciate from the rest of what I have to tell you, knowledge of the structure of RNA polymerase II forms a basis for understanding the entire transcription mechanism. Now, as I have said, when we began, there were no methods equal to the task. We had to devise an approach that was novel at the time. We decided, because X-ray analysis of large single crystals was not applicable, and no other methods were available, to turn to a simpler case, not of large single three-dimensional crystals, but rather to reduce the complexity of the problem by creating instead two-dimensional or single-layer thick protein crystals. The approach that we devised for this purpose and which proved successful is shown here. So we begin with a monolayer of lipid molecules shown in this picture spread at the air-water interface. The molecules are created for the purpose to adsorb the protein of interest through binding to the surface. The bound protein molecules will be constrained in two dimensions by the lipid layer, but they are free to diffuse rapidly in the surface, so in the plane of the layer, because of the rapid lateral diffusion of lipid molecules, a property of lipids and membranes which I had discovered myself as a graduate student in chemical physics in the 1960s. The advantage of this approach, the formation of these crystals requires an exceedingly small